Hello dear students, hope you all are doing great. So in this chapter, cell, the fundamental unit of life, so far we have studied about the details of prokaryotic cell, where we studied how a prokaryotic cell is so simple but well organized. Then we switched over to eukaryotic cell. First of all, we compared a plant cell with an animal cell, and we found out that there are many differences between plant cell and animal cell. Then we studied the membrane parts of the cell, where first we studied about the cell wall, and then we studied the details of the cell membrane. Today, now let's get into the world of cell inside and see how amazing is the cell machinery. So today we will study a very amazing but elaborate system which is present inside the cell, and that system is called as the endomembrane system, which includes organelles like endoplasmic reticulum, Golgi bodies, vacuoles and lysosomes. Why are they called endomembrane system? They are the part of the endomembrane system because their structure and functions are coordinated with each other. They are interrelated, intercommunicated. So let's study them one by one. We'll begin with the first of the system, that is endoplasmic reticulum. Endoplasmic reticulum is in a way a skeleton of the cell. It acts as a transportation system of the cell. So let's study the details of endoplasmic reticulum. The literal meaning of the word endoplasmic reticulum means endo means inside, plasmic means plasma membrane, and reticulum means network. So you can correlate that it is a network of tube-like structures which is present inside the plasma membrane. So if you see this is a cell, you can see this is the complete endoplasmic reticulum. It starts from the plasma membrane inwards and moves up to nucleus. So you can say it is a complete transportation system of a cell. And it has got channels. There are lumens inside which the things are secreted. Now let us go into the details of the parts of this endoplasmic reticulum. Endoplasmic reticulum may have these three parts. What are they? They are cisternae, tubules and vesicles. Cisternae are long parallel tubes which are stacked one above the other. You can see here, this is one tube, this is another tube. Such parallel tubes which are very less branched, they are called as cisternae. Cisternae generally have small outgrowths on them. These are ribosomes. You can see these black colored granular parts. These are ribosomes. So generally cisternae will have the ribosomes on their surfaces. And ribosomes are the protein factories. Now second part which an endoplasmic reticulum may have are tube-like structures. In fact, smaller tubules. That's why we say tubules. And these tubules are branched and they do not have ribosomes on them. So here you can see these are tubules. Apart from cisternae and tubules, endoplasmic reticulum may also have small structures which are called as vesicles, which are hair-like. So any endoplasmic reticulum may have cisternae, tubules and vesicles. Now depending upon how much is the percentage of cisternae, tubules, we can categorize the endoplasmic reticulum into two categories. One is rough endoplasmic reticulum, that is RER, and second is smooth endoplasmic reticulum, SER. Why they are called rough? Only because of the presence of ribosomes on their surface. They give a granular appearance. That's why they are called rough endoplasmic reticulum. Whereas in smooth endoplasmic reticulum, because they are mainly made up of tubules, there are no ribosomes. That's why they are called as smooth endoplasmic reticulum. Now let's see the functions. As I told you, most of the rough endoplasmic reticulum are made up of cisternae. They have more of cisternae. So they will have ribosomes on their surfaces. Because ribosomes are there, they are mainly responsible for protein synthesis. So rough endoplasmic reticulum will be mainly present on those areas, in those parts of the body, in those cells which are more concerned with the protein synthesis. Can you name few? Like our muscles, they are made up of contractile protein, actin and myosin. So they will be having rough endoplasmic reticulum. And these smooth endoplasmic reticulum, they are mainly responsible for lipid synthesis. So they will be more present in those areas which are responsible for metabolism of lipid. For example, in liver. In liver, most of the endoplasmic reticulum are of smooth type, smooth endoplasmic reticulum. And I just want to remind you, the sarcoplasmic reticulum, which are present in our muscles, they are also of smooth endoplasmic reticulum type. They will release calcium ions for muscle contraction. So there are certain functions which are common to both rough endoplasmic reticulum and smooth endoplasmic reticulum. First is they will form the network throughout the cell. So they are acting as transportation system. They are responsible for providing a particular shape to the cell. So they are acting as a skeletal system of the cell. Also, they are responsible for secreting so many substances. But the nature of secretion differs. The rough endoplasmic reticulum are mainly responsible for protein synthesis, and smooth endoplasmic reticulum are mainly responsible for lipid synthesis. So this is the difference. So the first part of the endomembrane system is endoplasmic reticulum. I hope you have understood. Now let's switch over to the next member of the machinery, that is Golgi bodies. The term Golgi body comes from the name of a scientist, Camillo Golgi. He was the first who observed these Golgi bodies in a cell. So the name is given as an honor for him. These Golgi bodies, I have written here Golgi apparatus and here Golgi complex. Golgi apparatus are the terms used for the Golgi bodies of plant cell. They are also called dictyosomes. They are simple structures and they may be many in number. But in an animal cell, the Golgi bodies are highly complex structure, and generally there is only one Golgi body, and that is called as Golgi complex. Now before going into the structure of the Golgi bodies, we will first study the function, then we can correlate the structure. The basic function of Golgi complex or Golgi bodies is it is acting as a packaging system of the cell. So whatever substance endoplasmic reticulum is forming, it is transporting to Golgi bodies. So it packs it and stores it. Second, it not only packs, it also does value addition, or you can say it helps as a processing system. How? Suppose endoplasmic reticulum has metabolized a specific type of carbohydrate and a protein. So this Golgi body has the ability to process it into another complex substance which is called glycolipid. Likewise, it can form more complex structures like glycoproteins, lipoproteins. These structures are basically responsible as receptors in the cell, or they may be the precursors for many hormones, and many other functions are there. So Golgi bodies and Golgi complex are a very important machinery of the cell. Now let's study its structure. If you see this diagram, you will see that the endoplasmic reticulum and the Golgi bodies, they are in very close complexity. They are very close to each other because the endoplasmic reticulum is responsible for adding up new structures to the Golgi bodies. You can see I have written two words. One is cis phase, other is trans phase. The forming convex shape structure which is facing towards the nucleus is called cis phase or forming phase. Whereas the concave maturing part which is away from the nucleus, it is called trans phase. So the endoplasmic reticulum adds on new structures at cis phase and Golgi complex processes it and releases for the use at trans phase. So this is forming phase, this is maturing phase. Okay. So Golgi complex is a very important machinery of the cell. After Golgi bodies, let's move to vacuoles. Next is vacuoles. Vacuoles are very simple structures. In a way, they are reservoirs or they are storing substances. So these substances may be water, it may be food, it may be different ions. Most of the time these vacuoles are much more concentrated as compared to the cytoplasm. Why? Because the storage is done actively by the use of energy. So last time I told you, so whenever energy is utilized, the transportation is against the concentration gradient. So most of the cells, they store the ions in the vacuoles. That's why vacuoles are hypertonic as compared to the cytoplasm. Okay, so these vacuoles are very large in case of plant cell. It may be generally single and occupies almost 90% space of the plant cell. And the membrane is called tonoplast. Whereas in case of animal cells, they are generally small, many in number, and they may perform many functions. They may store many food substances like starch, proteins or oil. So they are food vacuoles. They may be responsible for the movement, cytoplasmic streaming. For example, in case of amoeba, the movement is with the help of contractile vacuoles. And for providing the buoyancy, they may have gas inside them. So the vacuoles may be gas vacuoles. Okay. Now the last part of the endomembrane system is lysosomes. The last members of the endomembrane system are lysosomes. All these endomembrane system, they are single membrane. So is lysosomes. These lysosomes are just like vacuoles. they are formed from endoplasmic reticulum only, or sometimes they may be formed from Golgi body also. But they have specific types of enzymes called hydrolytic enzymes. These hydrolytic enzymes may be lipases for digesting lipids, proteases for protein digesting, carbohydrases digesting carbohydrates, and nucleases for digesting nucleic acids. So these are different types of hydrolytic enzymes. So depending upon their functional status, lysozymes or lysosomes may be of different types. They are primary lysosomes. They are having the secretions in them, but these secretions are not functional. They are in the preliminary stage. Then comes the secondary lysosomes. They are active lysosomes. They have the active hydrolytic enzymes in them. But the most dangerous are the autophagic vacuoles. These are special types of lysosomes which keep on searching for a faulty machinery inside the cell. For example, if a mitochondria is not functioning well, it has become old, so it will engulf that and digest it. That's why these are also called suicidal bats. They are scavengers, or they clean the system. In a way, they keep the system efficient. So these are the lysosomes. Today we have studied different members of endomembrane system. Now you have realized that all of them, they work in coordination and they are controlling a big machinery inside the cell, and that's why they are very important. I hope you have understood the whole system of endomembrane system well. Thank you.